Good morning, delegates to the Rumour SFAM conference on antimicrobial resistance changes coming. I hope, like me, you were able to join the first session this morning on companion animals and equine, which set the scene very nicely. I'm Nigel Gibbons. I was Chief Veterinary Officer for the UK from 2008 to 18 and had the pleasure of working with Rumour throughout that time, and indeed since, and I'm now a member of Rumour's independent scientific group. So I'm pleased to chair this session on aquaculture. Aquaculture is a new industry, really, and it is growing very fast and is one of the keys to solving the world's needs for protein as our population continues to increase. So harnessing that potential whilst ensuring animal health and welfare and protecting against antimicrobial resistance is really important. In this session, we're going to hear from two parts of the industry. We're going to hear from Dave Cockerill, who's Biology Director at the Scottish Salmon Company, about the salmon industry, its journey, the issues it's tackled, but the issues that remain to be tackled. Then from Professor Mathew Upton, who's Professor of Medical Microbiology at the University of Plymouth and Chief Scientific Officer at Amprologics, which is an antimicrobial discovery company looking for novel antibiotics and methodology to achieve that. So he's going to talk to us about work he's been doing with livestock, but techniques that have a clear possibility for application more broadly, including to aquaculture. Finally, we're going to hear from Oliver Robinson of the British Trout Association. He's been a trout farmer and a leader of the industry for a long time, being involved for 20 years or more, and has been involved in the industry as it's grown in the UK, including vaccine development in that industry. And he's going to talk to us about how the trout industry is tackling its rather different challenges from the much larger salmon industry and the challenges for the future. So without further ado, I'd like to hand you on to the speakers, and at the end of that, we'll return to a Q&A session.
Hello, I'm Dave Cockerill, the Biology Director and Veterinarian of the Scottish Salmon Company. I will present some thoughts on the management of bacterial challenges in salmon farming, a food animal sector that since its beginnings in the 1960s has undergone dramatic changes. In many ways, this compressed timeline that in terrestrial farming runs into centuries and millennia has accentuated the pitfalls and successes and taught some hard lessons that need to be applied by ourselves and others as we head into the uncharted waters brought on by climate change. From its humble beginnings in the 1960s, with the domestication of a few thousand fish, salmon farming is now global in its extent, producing 2.4 million tonnes on an annual basis. Scotland is the third largest producer, accounting for 8% of global production, with 200,000 tonnes per annum. This is dwarfed by the production of Norway and Chile, which together account for almost 80% of global production. However, this belies the importance of salmon farming to the rural Scottish economy and the value this production brings to the UK as a whole. There are 200 farms located along the west coast of Scotland and the Northern Isles, which, despite their broad geographical spread, could be condensed in surface area to take up less than half the footprint of Edinburgh Airport. There are 7 main producers, most of which are owned by large global salmon farming companies, due largely to the scale of investment that is required to successfully farm salmon. Salmon is the fish of choice for the UK consumer. As a nation, we purchase 64,000 tonnes per annum, valued at over £1 billion. The sector employs 2.3 thousand people directly and supports 10,000 supply chain jobs. As an industry, we contribute over £200 million to the exchequer and thereby make a significant contribution to the economy of the UK. A surprising fact for many is that Scottish salmon is the UK's largest food export by value, ahead of chocolate and cheese, and larger than beef and pork put together. It accounts for a value of over £500 million on an annual basis. As the industry expanded and the size and number of farms increased, there was increasing opportunity for pathogen transmission. Two microbial pathogens in particular became widespread during the 70s and 80s. Furunculosis, caused by Aeromonas salmonicida, was widespread in both Scotland and Norway, and cold water vibriosis or Hydra disease became established in Norway. This caused increasing mortality and decreasing growth and production, which threatened the collapse of the industry. In the short term, farmers and vets reached for antimicrobials to control infections. But by the late 80s, resistance to available medicines was commonplace. Research on the first vaccines began as early as the mid-70s, initially with limited success. To illustrate the scale of antimicrobial use compared to terrestrial sectors, I will illustrate this alongside the EU figures for pigs, poultry and cattle from the year 2007. At this time, many EU countries, including the UK, were using around 100 grams per tonne of production. Almost twice this level was being used in France and the Netherlands, and a lower level of around 20 grams per tonne in Scandinavia. At its worst in the late 80s, the Norwegian and Scottish salmon sectors were using around 1,000 grams of active per tonne of production. While this is historic, it is unsurprising that public perception of high antibiotic use remains. In fact, Chile continued even recently to have significant challenges with microbial infections endemic in their waters. The current position in Scotland and Norway is, however, completely transformed in this regard. Scottish figures in recent years have fluctuated between virtually 0 and 20 grams per tonne, while use in Norway has virtually ceased altogether. Not surprisingly, this about turn coincides with the development of effective vaccines against key pathogens. The first oil adjuvanted vaccines came to market in around 1990, and by the mid-90s the use of antimicrobials was negligible. Virtually all Scottish and Norwegian farmed salmon are now vaccinated for most or all of 6 key pathogens: Aeromonas salmonicida, Vibrio salmonicida, Moritella viscosa, Listenella anguillarum, and the viruses infectious pancreatic necrosis and Salmonid alphavirus. Alongside vaccine development, this positive trend has been bolstered by advances in fish husbandry practices and the veterinary services available to fish farms. Key principles to break cycles of infection are defined in the SSPO Code of Good Practice, to which all farms in Scotland ascribe. Essentially, Scotland is divided into farm management areas within which farms coordinate their activities to practice single-year class stocking, all-in, all-out stocking and fallowing practices, 4 weeks minimum contiguous fallow of whole farm management areas, and typically 8 weeks fallow of individual farms from final harvest to restocking. Veterinary services focus on prophylactic, proactive health management strategies with planning and surveillance to avoid and mitigate risks. Typically, monthly survival is around 99%, with few mortalities associated with bacterial infections. Turning to challenges of the future, there is no doubt that we are facing challenges brought on by climate change. Our peak temperatures occur typically in September of every year, and records show that this peak has increased steadily over the course of the last century. In fact, some farms in September of 2020 experienced temperatures of around 18 degrees steadily for several weeks. It is unsurprising that this would result in a shift in the balance between fish, environment, and pathogen. A significant change within Scottish salmon production occurred in 2012 when a condition that had previously been sporadic became endemic. Amoebic gill disease, or AGD, is caused by Neoparamoeba purulens, a protozoan parasite that lives on and damages gill epithelia. AGD is not linked directly to any increase in antimicrobial use, and no antimicrobial medicines are likely to have an effect on this parasite. However, there is no doubt that it has a profound effect on the physiology and resilience of infected fish. Tissue damage, loss of surface area, and the fish's immune reaction all lead to impaired exchange of oxygen and carbon dioxide, poor growth, poor feeding, osmoregulatory and excretory failure, and increased susceptibility to other infections. Highly effective treatment strategies for AGD have been developed in Tasmania, where this parasite has been endemic for several decades. Regular bathing of marine phase salmon in fresh water effectively kills the parasite. The Scottish industry is progressing well towards investing in the wellboat resources required to administer these treatments with sufficient regularity. In summary of the status of antimicrobial medicine use in Scottish salmon aquaculture, bacterial disease in Scottish salmon aquaculture is rare and sporadic. There is no routine or prophylactic use of antimicrobial medicine. Furunculosis caused by Aeromonas salmonicida is very rare, with around 100% vaccine coverage. Within the Scottish sector as a whole in recent years, antimicrobial use has varied from virtually zero to around 20 grams per tonne of production as an annual average across the sector. The Scottish Salmon Producers Organisation has set an ambitious target of less than 5 grams per tonne of production, which is 10% of the UK national food animal target of less than 50 grams per tonne. Vaccines, farming practices, and veterinary services have by and large negated the need for antimicrobials. Some farms, including all of the Scottish Salmon Company's marine farms, have not needed to use antimicrobials for more than 5 years. Looking to the future of antimicrobial use in Scottish salmon farming, antimicrobials are used for rarely occurring pathogens for which no vaccines exist, for example, Pasteurella caiensis. If such pathogens became more frequent, access to vaccines would be critical. Regulatory streamlining is required to allow rapid access to market for safe, effective vaccines. There needs to be a review of the cost of the regulatory process versus the commercial returns for pharmaceutical companies in low sales volume species like salmon. Typically, vaccines for salmon cost 3 to 5 pence per dose per antigen, which is little market incentive to vaccine developers. In terms of clinical disease, veterinarians have a duty of care to treat fish with antimicrobials if appropriate. Veterinarians need tools in the box, both medicines and vaccines, to secure the welfare of animals under their care. Currently, there are 2 available antimicrobials licensed for salmon in the UK. Low usage threatens the commercial viability of these medicines. There are limited options for product rotation or for treating emerging bacterial diseases should these arise. Climate change is likely to cause unpredictable shifts in disease risk as the balance between fish, pathogen, and environment is changed. Ultimately, consumer preference for antimicrobial-free production of food animals may drive practices that run counter to the pressures brought to bear by a changing environment.
Hello, I'm Mathew Upton. I'm a professor of medical microbiology at the University of Plymouth, and I conduct research into antimicrobial resistance. Today, I'd like to talk to you about the use of vaccines to reduce AMR and to present some data that we've generated on this topic. There's been growing recognition, I think, recently of the role of vaccines in reducing AMR, and a number of reports, including those shown here, have been published, but they have generally focused on human infections. And they've been argued on the basis that treating humans with antibiotics could be reduced if we vaccinate against pathogenic diseases. Having said that, the Chatham House report from March 2017 had a list of conclusions. The final one being that the issue of vaccine use and antibiotics in animals needs to be fully addressed. And the Wellcome Trust report, which focused on WHO priority pathogens in humans, does say that for Campylobacter infections in low and middle income countries, there are more studies required into the transmission pathways to see if animals really are a reservoir that's important for human infections. And if that's the case, then animal vaccination could be a preferred approach. And in the Nature Reviews microbiology paper, there's a section on direct and indirect use of vaccines on AMR. And it makes reference to the use of vaccines in food-producing animals to reduce antibiotic use and AMR, but it suggests that more studies are needed. One of the reasons that I have an interest in the approaches I'm discussing today is the co-location in Plymouth of the vaccine group, which was founded and is run by Dr. Michael Jarvis, a colleague of mine at the University of Plymouth. He's a virologist and he has an interest in viral vaccine vectors against hemorrhagic diseases like Lassa and Ebola. And Michael develops vaccines for use in animals to combat these and other zoonotic diseases. It was a while ago when you could actually go into work easily and meet regularly. That Michael and I had one of those chance conversations where you bounce ideas around, and we decided to work together, combining our interests in vaccines and AMR. And we're now taking a One Health approach to reduce AMR in humans by developing methods to vaccinate food-producing animals. So antibiotics are used less frequently in them, and that leads to less selection for antibiotic resistance in the environment, thereby reducing the selection for resistance. So we now have 2 programs running with a view to growing this activity. I have to say that our work together is not being developed in aquaculture, at least not yet, but there is a link. My co-panel members, I'm sure, will have much more to say on this topic. But Michael and I originally took some inspiration from reports on the impact of vaccines in the Norwegian salmon farming industry. The figure here indicates the reduction in veterinary medical products, VMPs, shown here, following the introduction of a number of vaccines over the 1980s and 1990s. So what's really surprising to us really was that although the veterinary medicinal product use went down, productivity actually went up quite a lot. So the yield of fish increased substantially over the following decades. Although these reports don't consider the impact of the vaccines on AMR, you can assume that a reduction in antibiotic or veterinary medicinal products in the water will reduce selection for resistance in the environment. So Michael and I have kind of taken the positivity of these reports and begun to apply the same kind of approaches in a number of settings where antibiotic use in animals has been noted as a problem. Firstly, in mastitis on the right here, this is a global issue where Staph aureus, Strep ubris, and E. coli cause an infection in cows, leading to a loss of yield and milk quality. The infection does require use of antibiotics, and that includes 3rd-generation cephalosporins, which the WHO have listed as being critically important for use in humans. And in 2015, the UK dairy sector used about 3 tons of antibiotic intermammary products to treat mastitis. There have been improvements since, but there's still a long way to go, I feel. There are some vaccines that are available, but they've got poor efficacy, and the microbial agents causing mastitis vary geographically. So we're developing a program to have a multivalent vaccine for mastitis, and I'll show some initial data on this in a moment. The other program we have is looking at porcine respiratory infection caused by Strep suis, and I'm sure we'll hear about this more tomorrow. The World Organization for Animal Health and the USDA have identified Strep suis as a major pathogen where a vaccine could reduce antibiotic use. So it's a major disease in piglets. So it causes 10% mortality in piglets, but it's also a zoonotic threat, and it leads to things like meningitis and toxic shock-like syndrome in humans. Probably the largest outbreak was in 2005, where there were 215 cases in China, and the toxic shock syndrome had a mortality rate of 62%. It's generally fairly well managed in the UK, but it does lead to large-scale use of antibiotics in some parts of the world, like China, where half of the world's pork is produced and consumed. Strep suis has a number of serotypes, 35 in fact. These three are the most common causing respiratory infection in piglets, but serotype 2 is probably the most prevalent and problematic. So there are some vaccines targeting these serotypes, but because they're capsule-based, the immunogenicity is poor, and because they're serotype-specific, wide coverage is also poor. So we're bringing a new approach to vaccine development for this disease. We have a new vector platform and a novel way of selecting antigens for the vectors. And so we feel this has real strong potential for use in the field with the potential for single dose efficacy. So I'll talk to you a little bit about that now. At the vaccine group, they use a range of virus vectors to deliver antigens. And we're using this virus, bovine herpes virus 4, as a vector for both the suis and the mastitis programs. And we've chosen this because it's been shown to be immunogenic in previous work, and it leads to a T-cell response as well as stimulating generation of protective antibodies. Although it's a bovine virus, it is permissive in pigs and cattle, so it will replicate, leading to expression of the target antigens, and that should then induce the required responses. This figure here shows where the target antigens are cloned into the bovine virus backbone, and they're cloned in replacing this ORF73. ORF73 has been shown to be important in latent infection. So cloning into this site and deleting ORF73 reduces persistence of the virus in the immunized animals, but it allows replication to occur and production of the cloned antigen that's then expressed. So with the levels of the responses seen in previous studies, the ability to have a multivalent vaccine with numerous antigens cloned in here, the low production costs and the efficacy, we think this makes an excellent vector. But the other innovation we're bringing to these programs is the way we select antigens to clone into the vector. So we're using reverse genetic approaches, and I've summarized those here for the Strep suis project. This has been used, reverse vaccinology has been used for other pathogens, but not Strep suis to date. And the principle really is that you can mine the genomes of bacterial pathogens for genes that encode proteins that could be recognized by a host immune response. These candidates can then be evaluated in the lab and then in animals to see if they stimulate a robust immune response. The issue with that, though, is that bacterial genomes probably contain generally thousands of protein encoding genes, and reverse vaccinology approaches allow you to filter out and focus on the proteins of interest. So the first step filters out proteins that are only exposed on the surface of the cell or excreted, because these are likely to be seen by the immune response and elicit a response. We then select proteins that cross the membrane multiple times again so that they're exposed to the immune system. We also want the target antigens to be pathogen specific, so that if an immune response is raised, it isn't directed at the host. And then the final step is to remove genes that have a high mutation rate. We're not interested in proteins that undergo random mutation at elevated rates, because this would probably lead to vaccine escape. And I guess this is very topical currently because of the discussions about coronavirus mutants that may evade vaccine cover. The selected antigens, so we've now gone from over 1,000 potential proteins in the core genome to, say, 5 targets. These are then given to the vaccine group, who clone them into the bovine virus vector, and they are then passed to our collaborators for evaluation in animals. And we're aiming to have a field trial, full field trial in the coming year, and assess the impact on antibiotic use. But we'd also like to assess the impact on AMR by comparing AMR in the environment in vaccinated versus non-vaccinated sites. The previous slide just gave a summary, really, an overview of the process, but there's more detail shown here of the pipeline that Katie Seeley has developed. Katie's a postdoc in my group, and she uses a wide range of software tools to triage protein targets. And she also makes reference to a number of publications, some key papers, listing proteins known to be surface exposed on Strep suis, and also studies that have revealed where some of these proteins are expressed in Strep suis when they're in an animal. So whether they're expressed in the brain, the heart, the lungs, those sorts of things. We also look in public repositories to see if there's any empirical data that these candidates have shown efficacy in vaccines before. So this allows us to prioritize proteins that will most likely lead to a protective immune response during infection. I'll finish my presentation just with some data from both of these programs. Here are data from the mastitis project where we've initially targeted E. coli, and the figure here shows the antibody levels in plasma that recovered from rabbits that were vaccinated with either wild-type bovine virus or bovine virus expressing our target antigen, designated target 1 here. So animals were immunized, and 12 weeks later, plasma was recovered from the animals, and that was reacted with E. coli cells to see if any antibodies reacted. And you can see from the figure here that animals that were vaccinated with the wild type had a much reduced response compared to those vaccinated with a target protein. In this table, there are some data on the Strep suis candidates that we've selected using the reverse vaccinology pipeline. So these are the top 10 candidates from about 1,900 proteins encoded in the Strep suis genome. For each of these targets, Katie will go in and she'll look at the distribution of B cell epitopes across the protein as shown in the blue line, and she'll also look at the hydrophobicity across the protein, and this helps us validate the candidates. So these are now being progressed at TVG, and they'll be evaluated for further triage and taken through to the field trials next year. Clearly, these approaches have much wider applicability, and some of the papers that are shown here include reverse vaccinology type approaches that have been used for fish pathogens. I should say that some of these pipelines are not as complex as ours, and a number of studies that suggest they've used reverse vaccinology methods start by selecting homologs of known extracellular proteins. So this means that proteins with hypothetical or unknown functions will not be analyzed, and that means that you'll potentially be missing some useful candidates. So that's a key benefit of some of the more complex pipelines that we're now using. So just to close with these summary points, there's growing recognition that vaccines should be used to reduce AMR, and we're developing approaches to do this in food-producing animals. There is an ever-expanding range of databases of pathogen genomes, and this provides a rich resource for development of sophisticated targeted vaccines. And the tools available to mine these genomes are continually improving. The approaches that we're using should be applicable to a wide range of bacteria, and you can also imagine approaches that allow you to only target the most pathogenic or virulent strains within a species, potentially with benefits to the host microbiome. We've shown that the bovine herpes virus 4 vector can be used to deliver antigens in animal models, and this results in measurable immune responses. So we're now following up with field studies to demonstrate that the immunized animals will be protected from infection with the target bacteria. And just to bring us back to the focus of this session, there is scope for use of these approaches in a wide range of animals, including fish, and I'd be pleased to discuss this if anyone is interested. So I'd like to thank the team in Plymouth who are working on our AMR vaccines, and I'd like to thank the organisers for the opportunity to speak at this great series of webinars. I'd like to acknowledge our funders and thank you for your attention.
Good afternoon. I'm Oliver Robinson from the British Trout Association. I personally have been a trout farmer since 1977. Aquaculture, and a big part of that could be trout, is the fastest way and most efficient way of producing protein to feed the nation. And I think that's one of the most important things that we can do. And I was going to give you an update on antibiotic use. And first off, really an update on the industry itself. BTA represents about 90% of the UK production, restocking and table farming. We have about 290 trout farms across the UK, employing well over 1,000 people. And you can see from our presentation, we produce about 15,500 odd tonnes. There's great hope that that will be increasing this year and 22 as well. When you have livestock, you have dead stock, you have disease. That is just life. It is fact. It is what happens in farming. We're all husbandrymen at the bottom of all of this. You know, that's our stock, and that's the way that actually if you have healthy stock, you've got, for us, if we have fish flow, we have fish health, and therefore potentially a financially viable industry. If we have a health problem, that can put our costs up considerably and cause all sorts of problems with bug levels on the farm. So the reality is that you're never going to have an antibiotic-free industry. That's not reality. Health issues we have demanding antibiotic treatments are enteric redmouth, eromonas, and RTFS. These all require the use of antibiotics. I will stress that there is no prophylactic use of antibiotics at all within the industry. And we have some very big concerns about availability of antibiotics, which I will touch on later on. I'd like to tell you a little bit more about the antibacterial usage since 2017 to 2019. We started in 17 collecting data from the farms, and that does actually represent about 95% of all trout production in the UK. So we've captured it very well, thanks to the vets who are responsible for doing it. And the mg per PCU in 17 was 20. 18, we were down to 13, and 19, we're down to 10, which is actually an amazing achievement. One of the reasons that has happened is there has been a huge concentration on fish health and welfare, change within industry from producing a portion trout, which is 300 to 400 grams, to producing a large trout, which is up to 3 kilos in freshwater and up to 4 or 5 kilos in seawater. With that change of size, the fish are now being injected at sort of 30 grams and giving them a much better cover. Whereas going back when we were producing a small portion trout that was sort of 300 to 400 size, we'd get about 8 to 9 months with the present vaccine. So that was a big issue then. So with the change of, believe it or not, the change of demand and the size of fish, that's actually helped us on the use of antibiotics. On top of that, we really have stepped up to the plate. Again, a great deal of help from the vets and veterinary medicines and CFAS and inspectors in Scotland as well, making people, staff aware of the issues. And look, you know, if you're going to use an antibiotic, you use it, but very, very carefully indeed. So that's a great success. I think our target is at 20. I can't ever see at the moment changing that, because a trout is cold-blooded. And when you have very hot summers and very high water temperatures, that can affect the bacteria loading in the rivers, and also obviously the fish at high temperatures are very vulnerable. Therefore, you can get more outbreaks of disease. So with climate change and more extreme weather situations and perhaps lower flows in the rivers, we're getting more vulnerable to bugs being around. What will antibiotic use look like by 2024 to remain below 20 mg per kilo across the whole trout sector? Realistically, the figure cannot reduce without more vaccines, and that is a big issue. We're a very small industry, and getting development of vaccines is a great frustration to us all, but hopefully in time we can get some. Less usage in the face of disease is a welfare problem. Antibiotics are there, if you like, as our guardian angel to make sure that we can treat and look after the health and welfare of our fish. There is already a widespread uptake of vaccines, and the BTA continues to support this work on new initiatives and on new vaccines. Achieving the targets. How are we going to do that? Make full use of the available vaccination programs we have, staff training on the farms, guided by the vets, and also working with the vaccine companies to make sure that when we're actually vaccinating fish, it's done in the correct and the proper manner. The vaccines can be administered in three ways. One, juvenile fish, so sort of 5-gram fish, which the immune competency of a 5-gram fish is actually quite low at that stage. And what we do is the vaccine goes into a solution of water and vaccine together, and the fish get dipped in that in oxygenated water for 30 seconds. And that does, the uptake is through the gills, through the skin. That does work quite well and give us maybe 8 months of reasonable cover. And I would say reasonable cover with the, not perfect, but reasonable with the, for 8 months. Then at that stage, you would have to either orally boost the fish, so I mix the vaccine in the food, and that gives you a shorter length of cover, maybe another 4 months. The alternative to that is actually injecting the fish at 30 to 40 grams, which will probably last the fish's life maybe 2 or 3 years, depending on what size of fish it's sold at, what side it is sold at. So those are the three methods of vaccination. We need veterinary expertise to review the strategies. So they're trained to look at that. They are separate to our industry, and they can come from it with a sort of fresh look at it, and also to make sure that the protocols are correct. Welfare training will be reviewed annually. There'll be training by the vets on the farms and also by senior management for the staff. And that will be from the very basic husbandry to how your fish are looking. For example, if you had sheep or cows in the field, if one was lying down and it wasn't looking very well, you'd walk up and have a look at that. It's similar to trout. Okay, they're in the water, but if they're swimming quietly or to the side of the tank out of the flow, you can pick up things. So from that basic understanding to being able to look for external parasites using microscopes for that, also taking bacterial samples, therefore cutting a fish open and taking bacterial samples for the kidneys, putting them onto a TSA plate, growing them, and being able to do the sensitivities. Each business will have a prescribing vet, and that will be on an annual visit. So if you've got a disease problem and you've done your resistance testing on it, and you end up with an antibiotic to use, but it's not licensed for use in trout, of which there is only one antibiotic, then under legislation, a vet can prescribe a drug for a treatment for an animal. Thanks to DEFRA, VMD and CFAS, and we've worked very closely with these people. They've all agreed, and actually they're providing a lot of help to do that. We'll be collecting the samples on the farms, i.e. the vets or the food company reps who do advise on fish health, or even myself if we have to. We'll be collecting some samples of the bugs on the farms and getting them down to CFAS, where they will be culturing those and running the resistance on the plates to see if that's good. And from there, we will then taking those bugs and also putting them into cryo, so we can hopefully we will be keeping them long term, so that in the future we may be able to get autogenous vaccines made for the industry, should the availability of other vaccines become a problem. The one antibiotic that we have a problem with or challenge with is oxalinic acid. And with new European legislation, it's become a sort of a big issue of whether we can use it or not. And to be able to use it, you have to show that you're using it responsibly, which we always do anyway. But by doing our resistance, by taking the samples down to CFAS, we can show that there is no resistance and no risk to human health, which is the important part. And then hopefully we'll be allowed to continuing using that antibiotic that's now an HBCIA. So i.e. it's gone up a category towards human medicine, and we have to be very watchful, very respectful, and very careful using it. But it is really the only effective antibiotic we have against enteric red mouth and furunculosis. The alternative to that will be oxytetracycline. To use that would increase, for that particular treatment, would increase our antibiotic usage probably by 7 or 8-fold, which is against everything we're trying to do. And it's not as effective either. So you could then get up resistance problems with the bug. So oxalinic is, we're going to have to keep that and work hard at showing responsible and getting advice from veterinary medicines and CFAS on how to use that. There've been no growth in resistance problems to oxalinic over the last 30 years. And it is a very important drug, as I said earlier on. We've been working very carefully with the VND and CFAS to see what can be done to use the antibiotics very responsibly and also get the resistance monitoring program going. The outcome, which is very exciting for us all, is that the samples will be taken on the farms by various vets and also some of the farmers who are trained, and those will be sent to CFAS, where they'll be able to do sensitivity on our main drugs. And again, that is a really important part to make sure there is no resistance, that we have no issues with it at all, but it's something we want to do in the future. As we then have these samples at CFAS, which are the bugs, we are hoping and will be creating a bug bank so that actually we could be using and developing autogenous vaccines into the future. So this is a very, very exciting program for the trout industry. We have various schemes running by Quality Trout in the UK. We established it in 2000. It's for the farmers, of the farmers, and it's all about the quality and the welfare of the fish, and very well recognised indeed. We have various other standards, Global Gap, Freedom Food, and various retailer schemes as well. We're audited every year, and as I say, also retailers have their own audits each year. So we're very regulated, which is a good discipline to have. Sometimes frustrating, but good. This is a British Trout. If you'd like to contact me, my details are on there. Thank you very much.
Hello again, delegates. I do hope you enjoyed those 3 talks on the challenges facing aquaculture and the in-depth one from Matt on possible new technology applicable to producing vaccines. We've got a lot of questions. So I've got the 3 speakers here with me. Dave Cockerill, Biology Director at Scottish Salmon Company. Matt Upton, Professor of Medical Microbiology at the University of Plymouth, and Oliver Robinson, Chief Executive Officer of the British Trout Association. Between them, we've got a wealth of experience and in-depth knowledge. So let's get to it. There's a lot of questions. So apologies if your questions don't get picked up in this session. Hopefully we'll cover the ground pretty well. And I'm going to use the thumbs up. So if you like a question, give it a thumbs up, it will pop to the top of the list. So currently at my top of the list is, beside vaccines, are there other preventive measures used in aquaculture like probiotics or feed additives? And I think I'll go first to Oliver and then to Dave on that one.
Yeah, good afternoon. Yeah, but in terms of alternative treatments, probiotics are and have been used over a number of years in our food, and they do work. They tend to treat the smaller fish, and then in a pulse system of treatment during their lifetime, and that certainly works indeed. And we're very keen to look to see what other treatments we could do to try and reduce our antibiotic usage.
Very good, thanks. Dave, anything to add from the salmon perspective?
Yeah, it's very similar. We do use nutraceuticals, immune modulation, probiotics, and particularly if there's any time where fish are exposed to a higher level of stress, which might be environmental or due to a handling procedure, these things are targeted and included in the feed to give as much nutritional support as possible when needed.
Very good. Okay, thank you. Really highlighting the importance of understanding your production systems and good management. Next question up is, how are antimicrobials or vaccines delivered to fish, and how effective are these methods? So we'll go in order of maturity of the industry. So we'll come to salmon first, and Dave, then Oliver on trout, and then Mathew from the perspective of these new potential vaccines. So Dave first.
Antimicrobials, when required, and it will be a very small proportion of the time due to the low frequency of use, but they will be included in feed. These are generally mixed at the feed mill at the prescribed inclusion rate, which will be defined by the veterinarian. And these the medicated feed is then provided to the farm for specific use on the farm under veterinary prescription. So it's a very tightly controlled process. With very specific amounts and doses being administered. As for vaccines, these are administered individually to fish by injection, intraperitoneal injection. This is done by hand generally still. There is an increasing move towards machine vaccination, where some of the robotic machines have become fairly sophisticated and very accurate. And increasingly with larger facilities, robotic injection systems are in place. Fish are anesthetized fully to immobilize, and then robotic systems will handle them and provide the appropriate dose intraperitoneally to the fish.
Really interesting, thank you. Oliver?
Well, we have a similar situation. Antimicrobials are used in feed treatments, very tightly controlled. The one thing I would stress, there is no prophylactic use from the trout industry of any antibiotics. It's identify the disease and only use the correct amount for treating that batch of fish. And very tightly controlled with our veterinarians. In terms of vaccines, juvenile stage for 5 gram, we use a dip vaccine at the hatcheries. And depending on the life of the fish, if it's going as a portion trout, sort of 400-500 gram, we would then boost it maybe 4 months later. And that's an in-feed boost. So you mix the vaccine in the feed and feed it to the fish. And that gives them another probably 4 months, 5 months cover. Beyond that, if we're going for a larger fish, they're injected intraperitoneal as well. And that's actually done, the fish are anesthetized, and then we have a team of people who are injecting actually an individual fish. It's not particularly mechanized at this stage, but I think innovation will allow us to do that.
Very good.
I think the development of vaccines is a key part of our future. That's a really important one.
Yeah, thank you very much. And between you, you're illustrating the different scales of the two industries and the resources that can be brought to bear to some of these challenges. But it's nice to see trout coming up and catching up with the same technologies. I'll just flag that people have the idea that people take buckets of antibiotics and throw them in the water, much of which drifts away. I think what you both said is it's in feed and all the feed gets eaten. So you've just busted a myth. So thank you very much for that. I'll go to Matt, just if you've got any points to make about emerging vaccines and how they might be administered.
Thank you. Yes, but I'll just pick up on that last point you raised there. I mean, I think depending on the antimicrobials being used, we should be aware of how much of that is actually excreted in a fully active form. So even though the feed is eaten, and that's why it's great that use is really low anyway. But coming to the specific question, I mean, that as you've seen from the talk, the vectors we use are viral based. So I'd probably be looking to identify a virus that is naturally present in the target population, and probably something like a herpes type virus, because we know that that they replicate, and hopefully one that can sort of super infect. So already infect a fish that's been infected with a similar virus. So that really we need to understand the biology of the virus that we're targeting. And so I'd assume that they're present in the water naturally, so we could be maybe looking at sort of, dip type approaches that Oliver mentioned there. And just going sort of slightly more off topic, but I mentioned Michael Jarvis in my talk. He's, as I say, a virologist interested in hemorrhagic diseases, and so some of these are in say gorillas and great apes, which are difficult to vaccinate. So what they're developing are disseminating vaccines. So you infect an animal with a vaccine vector, a viral vaccine vector, which can then transmit to other animals. So the animals infect each other with a virus which has an antigen in it to stimulate an immune response. So that way you can then immunize a small number of animals, and it disseminates to the whole population. Clearly there are sort of logistical regulatory things that need looking at there, but whether that would be applicable in these sort of settings, I don't know.
Very good, thank you very much. It may stimulate follow-up questions, which I'll keep an eye out for. Fish being so closely associated with their wild, with the wild species. But let's not dwell on that. I think it's important to look to the future. And aquaculture is such a big growing industry. Hopefully it will find room to invest in the way that the salmon industry has. Looking to the future, as the top question now is, what's your view on phage therapy? That's antibacteriophages, so viruses, rather than the use of vaccines to overcome use of antimicrobials. And I'll go first to Dave on that, and then back to Matt.
I'm certainly interested in phage therapy. We have a small number of farms in our systems, which are older farms, which it's harder to prevent pathogens from entering the system. When I'm referring to these are the I'm referring to freshwater farms in the early part of a salmon life cycle. And the future of that is moving to large recirculation units, which are extremely biosecure, and pathogen ingress is almost impossible. But there are still some freshwater farms where they are flow-through systems where pathogen ingress is difficult to prevent. And there are a few of these which have a background challenge with the odd opportunistic pathogen. I would be keen to look at phage therapy for that kind of unit, where there is a background challenge of an opportunistic pathogen coming from the incoming waters. What I have come up against is no particular regulatory framework which supports the use of phage therapy. There are some non-UK products which are available, but in order to import such products, they either need to be a vaccine or a medicine, and phage therapy is neither. So it falls into a bit of a regulatory quagmire at the moment, or just lack of framework. So I think there is some progress which has to be made on that front.
Very good, thank you. I'm sure our colleagues from the veterinary medicines directorate are tuned in, so I'm sure you've told them about this already.
Some time ago, we need to restate it.
Yeah. Matt, anything to add?
Yeah, I mean, the highly controlled environments, I think that's probably an ideal place for this sort of technology, really. Again, these are, unless they're engineered phage, I guess there'll be cocktails of natural phage that are selected to target the infectious agent of interest. So trying to contain it and have a regulated environment must be, I'd imagine that would be key to progress on the regulatory front. Yeah, and so I mean the only evidence I know of this being an approach that works in a clinical setting is in humans in kind of compassionate use cases where everything else has failed. But there is really good evidence that it's actually worked for some of those patients who are basically end of life and no other options exist. So this is quite a different setting, but it proves that the fact that they can be used in an effective way. Yeah.
Yeah, very good, thank you. And in this world of AMR, and when you depart from agriculture and look at the challenges facing the human field, no stone must be left unturned in getting at those bugs that are truly multiply resistant. Oliver, I'll segue slightly because there's a linked point here. You mentioned autologous vaccines.
Yes.
So there is a route to use that, but this is another old school technology. So do you want to add any more to what you said in your talk about autologous vaccines, how they might work?
Yeah, I'd be very keen to progress that and have a look at it. But again, we are quite constrained with the difficulty if you have one bug and that's taken from a farm, that effectively that farm can then use the vaccine for that's been made, the autologous vaccine that's been made. And a lot of the farms are very small. The vaccine runs are very big in terms of litres needed or produced to make it financially viable, and that gives us a problem. But I think over time, I see that as one of the routes that we could go. The difficulty we have with the trout industry is compared to the salmon, we're about 10% of the salmon size of the salmon industry in turnover terms or production terms, and therefore attracting vaccine companies to create vaccines for trout and help us is a problem because they're so expensive, and then you get a very slow return. And I think also, I know we've got our VMD colleagues on the line. I think long term, having a fast route vaccine system, a one-stop shop, if you like. I know we have to be very disciplined with it, but I think it's something that we do need to bring in innovation, and if we can have that system running. Somebody's come up with an idea, have a go at it and see if it works. And I think that's important. And yes, and autologous vaccines will play potentially a very big part of that.
Very good, thank you. I will come back to the questions, but there's such an obvious link to fast track approval of COVID vaccines and the use of technologies where you have a platform which is understood, where the safety of the platform is already established, that hopefully there is light at the end of that particular tunnel. Matt, do you want to add any more to that? Because this is your area of business.
Yeah, I mean, completely agree. Having sort of platform technologies there that you can flexibly use, so you can rapidly deploy a new vaccine by dropping in a new antigen. If the platform is accepted and validated and shown to be efficacious, then I think there's a good chance for speeding up regulatory progress. I think we need more research on animal models to validate new vaccines and help progress them more quickly. And clearly, the faster you can progress things, the more cost effective they are. And really, in a way, I mean, you mentioned that the fast track of COVID vaccines this last year. I mean, I think the economics of vaccine development is clearly a bit of an issue at the moment, but if it's more clearly tied to antimicrobial resistance and AMR in humans, then I mean that's a very valid justification for investing in research in some of these new areas. So it's about looking at the cost benefit models in a slightly different way. Yeah,
very good. Yeah, yes, it does need a push, but in the end, these companies do need to make a profit. So we have to see their way to getting a return. We're going to move sideways a little bit because one of these questions has reminded us that we talk a lot about bacteria, but the question is, does AMR include antiparasiticide resistance, such as that arising in sea lice infestations on farm salmon? Clearly it does. There's another question. There's a few on sea lice. We won't spend the rest of the Q&A talking about sea lice and parasites, but maybe I'll let Dave pick this one up and give us a comment on that.
Yeah, the subjects are interlinked. Certainly, antimicrobials are obviously not going to have an effect on either sea lice or gill amoeba, but both conditions have the potential to weaken fish and make them more susceptible to bacteria and opportunistic pathogens. I think that the case that antimicrobial use in salmon farming is generally very low, but then occasionally increases in an individual farm may need to treat because of an outbreak. These are usually sporadic cases where a stressed population has fallen a prey to an opportunistic pathogen. So there is a link and the need to control parasites, which is why I dwelled a bit upon amoebic gill disease in my presentation. In terms of sea lice, there is a drift away from using medicines for sea lice in any event. There are so many other methodologies for using medicines, for controlling sea lice. Mechanical removal, cohabiting cleaner fish, which are a biological control. These are highly effective methodologies, and generally speaking, the contribution of medicines to controlling sea lice has probably dropped to, I would argue, around 20% of sea lice control is medicinal, and the rest is through physical removal or prevention of infestation or biological control of sea lice. And yes, a common problem that affected the salmon industry when sea lice control was largely medicinal, and what still affects us in terms of bacterial infections is the limited availability of products. So you have relatively few products through which you can rotate, and that will drive resistance. So, for most of the 15 years between 2000 and 2015, there was probably 1 or 2 products available to most farms for sea lice control, and naturally that resulted in the development of resistance. There are more products now available and more methodologies available, and that helps us with product and method rotation.
Very good, thank you. There's a very quick follow-up, and it's so directly relevant. We'll take this now. How does sea lice resistance affect the use of in-feed and in-bath treatments?
I guess I was saying, the resistance to a product, there's no point continuing to use products which are where the effect is minimal, not least of all because of the extreme cost of using such a product for no particular reason. So that has driven the innovation of non-medicinal product methodologies for removing sea lice. And as I've said, I would argue 3/4 or thereabouts of sea lice removal is non-medicinal and therefore not subject to that process of resistance.
Very good, thank you. So there is an interesting question here. What is the contribution of fish food and terrestrial agricultural contamination to exposure of fish to antimicrobials? We'll take Oliver first, then Dave. Matt, I don't think this is your area, so I'll let you off. So Oliver.
Can you just repeat that? Sorry, a bit crackly on the line.
Yeah, what's the contribution of fish food and terrestrial agricultural contamination to antimicrobial exposure?
Well, I would say from our aquaculture side, certainly the trout side, minimal. We're mixing the food, the antibiotic in the food. It's been treated, it's been fed to the fish in a very controlled manner on specific feed rates, and we simply do not waste the food. And then minimum problems going out. And I personally don't believe there's a big issue with it. If there is any waste at all, certainly that waste is going to a very, very big, large volume of flow or water in the sea, and therefore diluting the situation instantly. So I don't believe personally there's a build-up at all.
Yeah, and the other part of the question is does terrestrial agriculture threaten aquaculture? So yeah, that would be from cattle, sheep to fish.
Yes, I mean it's certainly it is. I don't know. I mean that's an interesting question. In terms of antibiotics, I don't think there's a major problem there. And in terms of diffuse pollution in the rivers, slurry or something going on the fields and coming down the rivers and causing problems on the farms, and then causing stress levels on the farms and increasing the potentially having to use more antibiotics. Yeah, that is a problem, and I think it does need looking at. the diffuse pollution in agriculture is something we need to be considering very carefully indeed.
Yeah, very, very nice hospital path to terrestrial agriculture. Where your point is the dilution effect is so rapid and so great, you don't have a similar accumulation in the environment.
I personally, I'm not aware of it. We've done lots of work over the years of taking samples of fish from the rivers and things, and we've never found it yet.
Very good. Dave, do you want to add anything to that?
Yeah, I think just to add again that the use of antibiotics in fish is very infrequent, and this is not part of our regular strategy. It's an irregular occurrence for specific disease outbreaks in specific populations. It is within feed. It's not diffusely applied into tanks. Mathew made the point that some antibiotic is still excreted. Indeed, that is the case. So we can't assume that just because it's eaten, it doesn't potentially end up in the environment. But again, it comes back to the case that the use of antibiotics is very low and very infrequent. And again, if you were to compare that onto the scale of what is the use in some terrestrial systems where it's higher and perhaps goes into more concentrated water courses, I think the effect could potentially be greater in these sectors. But certainly from the aquaculture point of view, I do not believe that it's a significant environmental pollutant.
Yeah, which we could usefully contrast with the case with humans and the level of potential transmission through outflow of human use. The same reason, just there's a lot of us producing potentially a lot of outflow. Okay, Matt, do you want to add anything from a kind of science perspective to round us off, and then I'll go to the next popular question?
No, I'd probably agree with Dave and Oliver's comments there, really, in a way. I probably haven't got much to add, sorry.
Very good, no, that's great. A very particular point for Oliver, is the antibiotic use through the cascade system to be used frequently? Is it sustainable to use a non-licensed product for aquaculture? Oliver, I think this is repeating to you the problem that you raised in your talk, so you might want to expand a bit, but I think people heard what you said.
No, I think we asked that question quite considerably, because obviously if you're going down the cascade system, you've got something oxalinic, fluoroquinones and quinones. There is a concern getting towards human medicine with those. I can't stress more highly that the use of it is minimal in the industry. That's a really important one. And it's a very effective drug for enteric red mouth. In fact, it's really the only one that really does work. It is used specifically for one treatment, and then off we do our resistance test. So we grow the bug on a plate, we'll do our resistance levels on it. And since the last 20 years, there's virtually been with less resistance now than there was 20 years ago to it. So we're very responsible and very conscious as a sector for using that. Long term, it is a great concern with the legislation if things get moved up another tier towards human medicine. The other problem that we have is that we only actually have one licensed product in Trout for an antibiotic. We only have in total 4 that we can use. My belief is the cascade is used very responsibly and very safely and very carefully by our vets. Just to be clarified this, if we have a problem, I said to my talk, a problem with some fish, if you in a field, you're walking around, you've got a cow sheep lying down, you go and look at it and check it and maybe call a vet. We will do exactly the same if you've got fish, the same with the salmon swimming around in the cage or in tanks. You're looking at them, you're controlling your stock. If they don't look well, you constantly probably call a vet, get them in to have a look at it. Any morrow-bound fish, you'd anesthetize and dispatch, and then cut them open, take some bacterial samples from the kidney and grow them on a plate. So you know what bugs you're doing. We do get and a lot of our staff are all trained up to do that. So there's a lot of care in the use of antibiotics, particularly on cascade. That's really important to get that across.
Yeah, thank you. I think this question gets to the crux of one of the dilemmas with tackling antimicrobial resistance, which is, in the end, there is a need to use antimicrobials in the animal world. And I think both you and Dave may have made the point about animal welfare, but the strapline, as little as possible, but as much as necessary, has to be right here. And the other thing is, no food production system should be founded on the use of antibiotics. So you've both made the point that the use of antibiotics is exceptional in relation to a particular challenge. If we're there, I think there's a strong argument. Then you're looking at the problems, not only the fish species, where many species don't have enough drugs that are already licensed. So the cascade is absolutely essential. Dave or Matt, anything to add? No?
I'll just really quickly say that I think this is why it's really exciting that places like the Oxford INEOS Centre are suggesting they might try and focus on developing animal-specific antimicrobials for exactly that kind of reason. So I think that future is looking positive.
Very good.
The only thing perhaps to add is that, you know, as large as the salmon industry is, pharmaceutical companies consider salmon to be a small species and one which doesn't necessarily generate the returns that they would get for investing in products for cattle and sheep and dogs and cats. So I think it is important to understand that aquaculture remains, you know, a bit of an orphan in terms of where the pharmaceutical industry sees us.
Yeah, thanks. And from a terrestrial animal perspective, I think they in turn feel orphans compared to the human field.
Indeed.
With the challenges of new molecules being developed really for the human application, and then that's not antibiotics. And it reminds you of the O'Neill report that said there needs to be a stimulus to business that helps them, because the pure profit-driven motive isn't going to get you there. Vaccines might be better, because at least if you produce a really good vaccine, you have high volume sales, as opposed to a really good antibiotic you put on the shelf and never use. Further, there's one very specific question, because I love the question about the amoeba example. The bathing control of amoeba sounds expensive. What's the cost and what's the prospects for the future? This was Dave's point. What on earth is a well boat, and how does it work, and how much does it cost?
Well, a wellboat is a very, very expensive piece of kit. You know, we're talking tens of millions for some of these larger boats. Essentially, a wellboat, it has large tanks on it. Typically 2,000 to 3,000 cubic meters of water can be held, in which fish can be bathed. So it is very expensive. It's capital intensive, and these investments take time to get in place. So, and they are largely being made, and we're, you know, well on track to getting to the level which is present in Australia. The Australian control of amoebic gill disease is to bathe fish in fresh water every three weeks or thereabouts during the summer season, and perhaps less so in the winter season. We're not there yet, and we probably don't need to be there yet, because our water temperatures haven't reached the sort of levels which you experience in Tasmania. But ideally, I would like to bath all of our fish once a month if I could, and I'm not in that position to do so. It's not a very long bath. It'd be between 1 and 3 hours of exposure to fresh water, and it does them the power of good. But the logistical operation of getting them into fresh water, holding them and putting them back in their pens is expensive and time consuming. But you know, the returns are very clear. Fish respond very well to these baths, and amoeba are killed by exposure to fresh water. And so, you know, I think there's a very strong business case for this investment, but investment still takes time.
Yeah, what a great therapy, fresh water.
Yeah,
it's a brilliant example. I think we need to draw to a close now. You've reminded me to flag something that I found very striking from both Oliver and Dave's talks, which is climate change is having an impact. and disease challenges are going to go up, not down, even in this part of the world. The tropics are used to having huge levels of disease challenge. We're getting increasing levels. So the work of all of you to produce systems which minimize disease, and Matt and his colleagues to produce vaccines that allow us to systematically control disease, is absolutely essential. So in closing, thanks all of you. I hope the audience really enjoyed, I'm sure they did, both your talks and this Q&A session. And to everybody who asked a question that I didn't manage to get to, I do apologize. There were a lot of them. So thanks very much for all of your interest. Thank you very much, everybody. Goodbye, and I hope you join the sessions tomorrow.